Hello everyone. So as a part of the introductory neuroscience and neuroinstrumentation course, I would be explaining about how to do the EEG and ERPLab analysis. So there are various ERPs present. So for each ERPs, how to do the data processing using the MATLAB and the plugins like EEGLab and ERPLab. That is what I will be explaining in this demonstration. First I'll just give an overview of what is this ERPs. So what are the ERPs? ERPs are nothing but they are the event related potential. So our brain are having various various points are present in the over the electrodes. So the brain, it stimulates due to an event. There will be a stimulus that is being given to the brain, which based on which the response is being obtained from the scalp. So that is called as the event related potential. So based on an event, we are evoking a potential to be obtained. A biopotential is being obtained from that. That is called as the event related potential. So what are these kinds of events? It can be any kind of events. It can be a flash of light, or it can be a difference in the size of a ball, or different colors of balls, or it can be an auditory stimulus, it can be a tone, or it can be clicks. So these are the different kinds of events which we can be presented. So what is this AEP? AEP is the auditory evoked potential. So when we are giving a particular auditory stimulus, like it can be a tone, 1000 hertz or 500 hertz tone. So based on that tone, we will be getting the scalp records the potential based on the stimulus given. That is called as this auditory evoked potential. So the brain as such, it has various parts like the frontal, occipital, parietal, etc. So here the auditory stimulus, it is first recognized, or it is first generated in the auditory cortex, which is exactly above the ears, over the temporal lobe. So over there only we will be getting the proper auditory stimulus, the response for the auditory stimulus. So here we are using an experiment. In this experiment which I have been working, we were using the Neuroscan 64 channel EEG system, wherein the cap, as I have shown over here, the electrodes are being placed over the head, and these electrodes, these points over here, these are they are the electrode which creates contact with the scalp of the head, and that is how the EEG is being acquired. So first, for any step before the data processing, we have to first acquire the data which has been obtained from this Neuroscan EEG system, and the stimulus is being given through a software called as a STIM software. So where we can create whatever kind of stimulus which we want. For example, in this case of auditory evoked potential, we require a auditory stimulus or a audio signal which is 1000 hertz tone, which we have used in a frequency of 1 hertz, which means for a second there will be a one signal that has been the one trigger will be given by this system to the acquisition Neuroscan system. So these triggers are given, and the tones which can be, these tones, it can be presented in whichever manner. It can be through the headphones or by speakers. So in this experiment, we had given the AEP through the speakers, and followed by in the Neuroscan acquisition system itself, we can assign various parameters. For example, we can give whichever, if we want 64 channel, that accordingly that configuration we can give, or it can be a 32 channel configuration, or even if we want only the frontal electrodes, we can give just 3 electrodes for the forehead, just forehead bands. So like that we can give the configuration can be given in that manner. So then we have various other, like during the acquisition itself, while giving the stimulus or by giving while giving the triggers, what happens sometimes we can't be in a stable position for a long time, so we can change our there will be a muscle movement, or we just blink our eyes, or there can be different kinds of artifacts that can be produced during the experiment. So those artifacts can be rejected using the using a voltage threshold from -100 to 100 microvolts, because mostly all these blinking of the eyes or any other muscle contraction or the EMG artifacts, whichever artifacts that has been produced, it can't go more than 100 microvolts. It will be in that range only, because our EEG signal is like it comes in very few microvolts, and over that any artifacts will not go beyond -100 to 100 microvolts. So for that reason we keep a voltage rejection threshold, and reject these in this range. We will reject the signals, and then we can there will be various other noise artifacts or this EMG artifacts and all, which can be removed by means of filters. So we can have a notch filter or a band pass filter, and a band pass filter both together. If you use, you will get a proper ERP. Now whole of this EEG, it's like a continuous data. It will take about maybe 2 minutes or 3 minutes of the experiment, or it can be even more. For AEP specifically, it will take about 2 minutes at most. So after that, but our interest is just to get a particular time period, just from a few milliseconds. So like the first initially some 500 or 300 millisecond, along that range only we require any ERPs. So we can just cut down or cut down that particular part of the that particular process where we cut short the time period according to our interest. That is called as epoching. So I'll just give you how does this AEP looks like. So this is how the AEP actually looks like. Here this P1 N1 P2 complex, that is the that will be present in any auditory or visual evoked potential. In all the evoked potential, this particular complex, this N1 P1 P2, this complex is called as the late complex. So LPC complex, we say that as late positive complex. So this particular this particular N1 that is there, that is the most prominent peak that will be obtained in the 100th millisecond of recording. So this particular, if this is there, we can confirm that our auditory the auditory cortex that is there, it is properly functioning in our brain. So this particular here this red and blue zone, this blue color area, that is called as the primary auditory cortex, where the evoked potential will get activate, or it will generate the AEPs are generated in that region specifically. So after this data acquisition from the neuroscan, we will get a continuous data, like as I told you, it will be from it will range from for about 200 seconds, or it can be more than that also. So so that particular we have to further process it using this EEGLab and the ERPLab. So I'll just give you a small overview of what is this EEGLab and this ERPLab. So this EEGLab is nothing but the it's actually a MATLAB toolbox that is actually given by the Schwartz Center of Computational Neuroscience. So these particular they have given a plugin to just process the EEG and the ERPs basically. So EEGLab is like to import basically EEG and ERPLab, there are two different things, but they work under the same user interface. So this gives instead of scripting, some people will have difficulty in scripting. For at least for one data, we can give graphical user interfaced methods rather than scripting. So using these EEGLab itself, we can have a we can make the scripts according to the function we do using the user face itself. So this MATLAB toolbox, it helps, it's having these graphical user interface approaches, and then we can have various EEGLab data formats. Now EEG acquisition, it need not be just in form of PDF or CNT. There are various formats that are there, and this EEGLab, it is having different plugins also, with which we can import all those different kinds of EEG data formats, and then we can have various plotting methods, and we can have the artifact being removed, and we can have the different time topographic analysis can be done, and various other advantages are being there, being user friendly also. It has been very useful for, it's like a very basic step for EEG analysis. And then there are various other plugins available, like in order for example, if you want to do some other pre-processing steps, or if you want to do some other artifact rejections, or various other formats in which we can import, export the pre-processing steps. So like that, there are various other extensions that is available, which I will show in the demonstrations, like how to get all these plugins installed and how to go about this working with this toolbox. So this is just the primary, like for importing alone, this particular data toolbox is being used. Next we will have this ERPLab. This particular ERPLab is also a free source MATLAB package itself, which helps in analyzing the ERP data. So the whole of EEG is like a continuous whole data. So here, for example, this green color, the green waveform that has been shown over here, that is the whole EEG waveform that has been there. But for us, this red color here, that is called as the stimulus. For that stimulus, we want to get how the brain responds. So at least just a few seconds or few milliseconds of the data is only required for processing our for getting the ERPs. So we can do that epoching for a particular time domain alone. So for example, I just require from -50 millisecond to 200 millisecond in order to process the AEP analysis. For the auditory evoked potential, we just want that LPC complex to be seen. So for that reason, we just require maximum of 200 millisecond or maximum of 300 millisecond, not more than that. So we will take a small baseline from -50 from the trigger. We just don't require the EEG or the ERP waveform from the trigger. We just want just a few microseconds before the trigger starts. We wanted the ERPs to be analyzed, so that we will get an idea of how the waveform looks prior the triggers being given. So this particular step is called as epoching, and this is a very basic step for a ERPLab as concerned. So for this I will again show you how it looks, how to do the various the steps for AEP with this particular EEGLab and ERPLab. And these sites I have mentioned over here in this place, where we can in this sites we will be obtaining all those EEG and ERPLab zip files we will get, and we can download it, and how to import that into our MATLAB, that also I will be giving you as a demo. Yeah, so these are the demonstration steps that will be that I will be covering. So firstly I will so how to import the data, then how to do the filtering, then how to scroll the data along the, how to see the whole data, how it looks, and then how to create this event list. Like the brain as such, you find that this triggers, it is given from the neuroscan system, but then what happens is we wanted we want we can't just take the triggers as such. We have to assign a different name for that. So how to do that and all is the next step. So how to epoch the data, like as I said, we wanted to be in this particular time frame. So in that, how to take that epoching, and how do we reject all the artifacts. As I told you, there will be various artifacts of blinking and muscle congestions, or when we move our head, or we can't be still for while taking a data or while acquiring a data. How we can't just for 2 minutes we can't be steady like that. So there will be a movement in our head, or there will be some muscle movement, eye blinks or anything. So that can be rejected using these artifact rejection in EEGLab and ERPLab. And then we have other filterings like band pass filter. So this is the sequential order in which we have to do any of the EEG or ERP analysis, and then finally we can obtain the data. And another one important thing about this ERPs is that almost always all the ERP, all the evoked potentials, it will be in the form of a negative up, like always as I shown in the figure, there will be always, it will be always in the negative up manner. It won't be most some P300 for example, those and all will be in the positive positive up, into we can obtain in the positive up manner, but otherwise almost every other all potentials will be in negative up only. So I'll just start up with the demonstration of how it looks in the actual live processing. How do we do it? I'll just go through that. So these are the different references that can be useful for this particular presentation. Like we have the different ERPs and how is it characteristics and how does it look and how it has been started working, how they started working. Everything has been explained in this paper, in the first paper, and the next two are the just a base paper about the EEGLab and the ERPLab, how the software came into picture and how they have done all the steps regarding it. So that has been given in these two papers. So the next I will be giving you a short presentation, so a demonstration of how to do the AEP analysis, and then followed by I'll give the other next like this AEP. There are various other ERPs that are there, like the P300, MMN, VEPs. So along with that I will give their presentations and followed by its demonstrations as well. So thank you, and I'll start with the demonstration now.